Okay, welcome to the April Cannabis Cultivation Webinar. We will be starting shortly, but first, we would like to allow a brief moment for attendees to join the webinar. It seems like the number of attendees is increasing, so we can wait another few more minutes, a few more seconds. Okay. Maybe I can begin now. Okay, hello, my name is Saint Park, senior scientist at the ICR, the Institute of Cannabis Research, and chair of this webinar series. So ICR and Volcani Institute in Israel are pleased to have you join us for the April webinar. Before we begin our presentation, I would like to inform you that the ICR will be hosting the ICR Cannabis Research Conference in Denver, Colorado from August 3rd through 5th. If you are interested in participating, please visit the conference homepage or contact ICR directly, and we will be happy to provide you with more information. Also, I would like to provide some information regarding the logistics of the webinar. Today's presentation will be followed by a short question and answer period. To ask a question, please use the Q&A feature in the Zoom toolbar. You can enter questions at any time during the presentation, and we will address as many questions as time allows until the end of the hour. Please do not use the chat function to pose questions for the Q&A period. Instead, please reserve the chat function for technical issues and questions. Both the Q&A and chat functions can be found in the toolbar on your Zoom screen. It is now my pleasure to introduce Dr. Meiri Bernstein, Senior Research Scientist at the Volcani Center, Ministry of Agriculture, Israel, and co-chair of this webinar series. She will be introducing today's presenter and moderating the webinar. Meiri.
Thank you, Sang, and welcome whoever joined, all of you that joined us. Good morning, good afternoon, good evening, depending wherever you are. I'm honored to present today's speaker, Professor Dedi Meiri, and we're very thankful and grateful for his willingness to share some of his knowledge and information and data in our webinar series. Professor Meiri heads the Laboratory of Cancer Biology and Cannabinoid Research at the Faculty of Biology in the Technion, which is the Israel Institute of Technology. Dr. Meiri comes from a highly diverse scientific background in biochemistry, in plant biotechnology, and in cancer. And it will be really wonderful to see integration of some of these aspects in today's talk. The research which is conducted in Dr. Meiri's lab sets out to bridge the gap between herbal and modern medicine, and the lab investigates the vast therapeutic potential of secondary metabolites from cannabis, as well as from other herbs, from algae and psychedelic mushrooms. And it reveals how these naturally occurring metabolites affect various types of cancer, inflammatory diseases, and disorders of the nervous system, such as epilepsy, Alzheimer's, and others. The very active lab of Dr. Meiri consists of highly trained team of skilled professionals and students working in synergy to achieve the highest level of results to better understand the complexity and diversity of cannabis and other natural compounds, and realize their full therapeutic potential for human health. And I'm very much looking forward, Dedi, to hear your talk today and the interesting data that are coming up from your lab. So please.
Okay, thank you very much. Hi, everybody. It's a pleasure for me and an honor for me to be here. Thank you, Meiri, for inviting me and hosting this webinar. And thanks for the audience for taking the time to joining us. As I was presented, I have a laboratory that doing cannabis and other herbs research and potential of affecting human diseases. I want to do just a short disclaimer saying that I also having a company called Cannabinoid that giving services to my lab and other companies. And part of the results that I will show today was also been done at Cannabinoid. So I'm assuming that most of the audience here knows these flowers and know what it is. And I don't need to start to talk about the history of cannabis and the regulation situation, but I do want to give a short introduction saying that actually this flower, the cannabis, is a unique flower because it's harboring a unique molecules called cannabinoids. And all of you probably know that we see here 5 major cannabinoids. But this plant is very unique in affecting our body because it's actually affecting a system that is existed in our body, what we call the endocannabinoid system. And this is a unique system because every cell in our body, we have 220 some types of cells in our body, every cell in our body harboring receptors for the endocannabinoid system and can produce endocannabinoids. So actually every cell in our body being affected by the endocannabinoid system. It's a system that regulates and do modifications and balancing other systems and the cells in our body. And I won't go into that. I will just say that this is actually a system which is a communication between cells. One cell sends a signal to the other cell to do something. Increase appetite, reduce feeling of pain, symbiosis of glucose, everything that cannabis is, or the endocannabinoid is regulated being by signaling between a molecule called endocannabinoid, the receptors in the other cells that capture that. Cannabis is just mimicking that by creating these molecules, the cannabinoids, that can bind the receptors and send the signal, affecting the immune system, affecting nerve system, affecting cells that creating insulin and the appetite, the feeling. And I can go hours about talking about all the mechanism of action in our body and why these molecules affecting them. But actually, if I will ask you where cannabis is growing in the nature, and since we can't really speak in this situation, I will ask it as a hypothetical question. And usually people will say it's originally from Nepal, it's originally from India, Siberia, I heard, in Western Europe, I heard many answers. But then usually I'm asking a more simple question. If you're talking about dogs, let's say a poodle dog, okay? Where poodle dog is growing in the nature natively, and everyone knows that there is no poodle dog in the nature. It's actually, we're creating these from breeding wolves. Wolves started to help humans 10,000 years ago and even more. When humans started to hunt, the wolves started to go around them and eat the leftovers. The humans pay attention that the wolves protecting them from a worse animal like tigers or other animals. So they started to feed them and started to breed them. So they breed the one that look more nicer, more friendly. And after thousands of years of breeding, we got these ones. But this is not a native plant. This is not a native dog. This is something that we're creating. And cannabis is quite similar to that. Cannabis is the oldest plant that we know that human beings did breeding and grow in agriculture. And for that reason, we don't have actually cannabis that's growing in the nature as a native plant. It's all breeding and breeding and breeding that we did. And for that reason, probably we create a plant that harboring huge amount of these active molecules, the cannabinoids. If we're looking on other plants, we know other plants that harboring cannabinoids, but the percentage is extremely, extremely low. In cannabis, the percentage is so high, probably because humans breed it to be like that. I'm not sure that it's helping the plant to harbor 25 or 27% THC or 20% CBD in the flowers. It's something that we created. But cannabis, it's not a name of one plant. It's a name of many types. We call them chemovars. And in my lab, only I have more than 1,000 different chemovars of cannabis, which each one of them will contain different combination of these molecules, of these cannabinoids. And if we look on a slide that we made of 20 different cannabis extract that a patient can get in Israel, it's flowers, not extract. And if you look every line, it's a different types of flower that patient can get in Israel. This is from 2018, so it's quite old already. And the rose is actually, when it's black, the meaning that these phytocannabinoids is highly expressed in that plant. When it's white, the meaning that it's not expressed at all. First of all, you can see that there are many phytocannabinoids. Part of them, you know, probably THC or CBD, but there are many other cannabinoids. We can analyze today 160 phytocannabinoids. But what I want to show you that even though, let's look on these two in the blue line, even though both of them are high CBD strain, there are many different molecules that express in one of them and not expressing in the other, or in the other one. And there is a big diversity of these molecules in the different plants. Today, we can analyze or can identify 11 families of these phytocannabinoids in the plant. Altogether, we can analyze a little bit more than 160 phytocannabinoids. You know that there are other active molecules like terpenoids and flavonoids. So it's a combination of a lot of these secondary metabolites, the cannabinoid, terpenoid, flavonoids, that are working together to achieve an effect on our body. And as I said, we can divide these groups of cannabinoids in different families, but you can see that one group is a line of cannabinoids that we know the structure and we know how they look, but it doesn't even have a name. We call them just by number, when how we can identify them. And there is still plenty of phytocannabinoids that sometimes express in high levels, in 1%, 2%, 5% in the plant, that we don't have even a name for them. We know they are cannabinoid, we know they are active, but we don't know exactly what they are. So we can take all these cannabinoids, and we did, analyzing thousands of samples, and we can divide them actually in 4 major families. Ethan Russo published, I think it was 2007, when he named this group. He said, every cannabis that have high THC, it will be type 1. Every cannabis that will have high CBD with low THC, we call it type 3. What we call balanced plant, which have high CBD and high THC, we call them type 2. This is the purple one. And in a publication that we published 2 years ago, we identified another types of cannabis which have high CBG, but very low THC and very low CBD, and we call them type 4. But what I want to show you here, that we can identify molecules which are totally unknown. We know it's cannabinoids, but we don't know anything, but we can see that it's appearing only in type 1. Even if I don't know how much THC I have in this cannabis, just looking on these molecules, I know exactly that this is a type 1 cannabis. The same thing For the CBD group, this molecule 3318A that I will show result in a moment on this molecule, which always came in type 3. So even though it's a whole mess and there is many molecules, still there is a meaning for saying this is high THC, low CBD, or high CBG, low THC, to have a group of molecules that working together, probably because of their biosynthesis. So the biggest question in our field always was how to match the right flower to the right bride, how to match the right cannabis to the right indication. If somebody is suffering from pain, what will be the best cannabis to reducing pain? If somebody have seizures, epileptic seizure, what is the best cannabis to reduce seizures? If somebody suffer from autoimmune disease, what is the best cannabis to help this autoimmune disease? So what we're doing in our lab, we are doing this study and trying to identify the molecules and understand the mechanism of action. I will give here 2 or 3 examples, but the examples for themselves are less important as the idea that I want to show in my talk. So how are we doing this research? And we can grow cells from our body, let's say cancer cells, in a plate and test the cannabis of them. We can grow them in a plate that have many holes, many wells. Let's say these called 96 wells. We can grow, let's say, prostate cancer cells in this plate, and in each well to do different experiment, and then to look which cannabis affecting the best. We have equipment that can do it automatically very fast. For example, this is called high throughput screening. This is a microscope that can take images from each well and to screen that. You can take 1,000 images an hour, and if you put the plate for 24 hours, and this is live cells, you will get 24,000 images, and then the computer can calculate and can tell you in which well you get the best effect. It depends what effects that you're looking, killing the cells, blocking their migration, blocking secreting glucose, whatever you're looking. So these are results from this work. This is Eli Besser, a Ph.D. student in my lab. This is a work that was actually accepted 2 weeks for publication. So what we're looking here is Eli checked how cannabis affecting the cancer of the immune system, what we call leukemia. And this is leukemia called TLL. So you grow the cells in a plate like I showed you and put many types of cannabis. We're looking now at 15 different extracts, but actually we tested more than 250. So we're taking different types of cannabis, making an extract, and put on the cells in these wells and check how it's affecting. What we see in the graph is the viability, how many cells survive and how many cells die. So if the graph is lower, more cells die. So you can see that in 2 microgram per ml of cannabis, of whole extract, most of the cannabis didn't affect these cells, but there was one extract that killed these cells very efficiently. So we ask, okay, leukemia, it's a given name to different type of cancer. There is AML and TLL and CLL. Whether this extract number 12, is efficient for all the types of the leukemia or very specific. And we saw that 50% of the leukemia, it didn't affect them at all, but 50% of the leukemia, it's killed them very efficiently. When we test genetically, we saw that in all the cancers, all the leukemia that the cannabis is killing, there is a specific mutation in a specific protein called Notch1. Every time that there is a leukemia with Notch1 mutation, these types of cannabis will kill this cancer. And every time that there is no mutation, it won't kill. I want to show you how specific it is. Probably a lot of you know what is Rick Simpson oil. When we taking cannabis, making a very high potent cannabis extraction and try to treat cancer. But if I'm showing you these results, if I'm going backward, what is the possibility that you will hit the right extract from the 250 extract that really killing this cancer? And more of that, it's not for all the leukemia, it's specific type of a specific type of the leukemia. It's what we call personal medicine. In 50% of the leukemia patient, there will be this mutation. And for that reason, 50% of the leukemia this extract is killing. And I won't go deep in the biology, what is this mutation and what is doing on the membrane. I will just say that we took these cells of the cancer from humans. We create this cancer in the mice. You can see a CT here with a tumor in the mice harboring this mutation. And now we treat these mice with this cannabis, and you can see here the size of the tumors that are not treated, which growing exponentially, while in gray, the tumor that have been treated with the cannabis, and you see that the tumor are not growing at all. This is the stat of the mice, and these are the tumors before and 3 weeks treatment. Because this is leukemia, we did a bone marrow implantation, we saved the mice life. We actually did 5 different types of mice model to prove that this extract is really working. But now, the real question, these are all the cannabinoids that this extract is harboring. You can see here high CBC, it's a 50% CBD in the extract, but look how many compounds, how many cannabinoids, look how many cannabinoids are even doesn't have a name. They're just numbers, even though sometimes it's expressed nicely. Here it's 1.2% of these cannabinoids. So now we're asking how or which cannabinoid are doing this effect. And the way to do it, and the way we're doing it, is by trying to limit it. So we're taking all the extract, this is an HPLC graph, and we fraction it. We're taking all these cannabinoids here in one tube, all these cannabinoids here in second tube, and so on. And we're taking these pieces of this extract, we are actually cutting it, the 100 and something cannabinoids in groups. And we are going back to the cells and to the mice, and we saw this is the control, this is the whole extract, killing the cells very efficiently. And here are 4 parts of this extract, and you can see that fraction number 2 was very efficient to kill all the cells as the whole extract. So all the cannabinoids that were here, here, and here, we can eliminate, we don't need them. And we're going back and forth, and I won't go deeper to that until we succeed to analyze 3 cannabinoids, that if you're taking the 3 of them, isolate them, purify the 3 of them, and put together, you get exactly the same action as the whole extract. But every one of them or 2 of them together is not working. You must have the 3 of them in order to kill the cells. And now with 4 years of a lot of biochemistry and cell biology, and I won't go into that, but we did a lot of work, we succeed to identify exactly what are the cannabinoids that are doing this effect. Just a moment. What are the receptors they are binding? They're binding CB2 and TRPV1. What are the action in the cell? What they are activating in the cell that create a death in the cells that have Notch1 mutation and not affecting the cells where there is no mutation. So what I'm trying to say, without going to the biology in deeper, that today we know exactly what are the molecules, but also how they're doing this effect. What is the mechanism of action? And when I'm speaking with a physician or with a scientist, I can explain him why exactly it's doing that effect. It's not something that is unexpected today, and we can understand that. So this is one way how to start something that you don't know anything, and starting a screen and reach to the molecules that are doing that effect. Another way is to take knowledge already from patient. Between 2015 to 2020, my lab did a lot of database on the patient in Israel. We actually collected data from around 20,000 patients where we know exactly what are the cannabis they are doing. We're doing all the analysis of the active compounds and trying to measure that with the output of the patient. And what we learned from that a lot of patients telling that the major output, the major reason they're using cannabis, it's because it's helping them to sleep. So here, we didn't need to screen a lot of extract. We know from these 20,000 patients, what are the major cannabis, what are the major chemovar types of cannabis they are using that help them to sleep. So we pick 10 of these cannabis extract to start this screening, and we defined one extract that's working very well on the mice, make them to go to sleep very well. So we have cages with the mice. The mice is actually going to sleep when there is a light and awake when it's dark. And we have camera that following up on these mice. And we make these mice feeling tired, and then we turn on the light, this stimulating sleep. Sorry. And these mice are we can measure how much time it takes them to fall asleep, how many times they're waking during the sleep, what is the length of the sleep, and we can also put electrodes on them and measure EEG and see whether this sleep is efficient or not. So we define one of these extracts that every product that we did from him affect different parameters of sleep. It's make them to go sleep faster, but also the sleep was very deep and very efficient. And we tried it with these different batches. You see, this is the control, and the mice here that getting these types of cannabis going to sleep faster. We measure the EEG in the brain of the mice, and you can see here, this is the time that the mice was awake or feel awake during the hours that he is sleeping. So a mice, every 2 minutes, lifting his head and looking around whether there is no cat around. So this is something evolutionary. Every 2 minutes, he's kind of waking up for a few seconds and going to sleep. So you can see here the awake time of a normal mice. You can see here what we call NRAM and the RAM sleep. And the mice that got these types of cannabis, you can see, first of all, if I'm going backward, look on the red one, the awake time during his sleep is reducing dramatically. The NREM is growing dramatically. The REM is disappearing totally. So we see that we're really affecting the sleep in the brain action in the mice. But again, the same question, we have many molecules in this extract. This is an high THC. So how we define which molecules causing the mice to go to sleep? We can do the same thing. We can cut these molecules to partial pieces. We can take this extract and fractionally take all these cannabinoids in one tube, the THC alone in second tube, and all these molecules in third tube, something like that. And when going back to the mice, and we are very surprised and happy to see that actually we need just this piece without the THC and without all these cannabinoids in order to make the mice to go to sleep as good as the whole extract. This is the control, this is the whole extract, and this is just the piece from the left. And we're looking what we have in the piece of the left, on the left, and we see that there is quite a lot of CBN and CBG and others. So we're cutting now the CBN out because people know that or think that CBN can make you tired and sleepy. So we're cutting that CBN out. Now we have this piece, something like 20 cannabinoids without THC, without CBN, and it's still working very well. Actually, we succeed to reduce it to just 2 molecules, both of them unknown, that if you give these 2 molecules, we call them GTS1 and GTS2, go to sleep 1 and go to sleep 2. These are cannabinoids with a structure a little bit similar between, I will say, it's similar to the THC and the CBN, but different chemistry. We call them GTS1, GTS2. This is unknown cannabinoid that if you take the 2 of them, just the 2 of them, the mice will go to sleep as good as the whole extract. And now we have actually 2 pathways. We can take these 2 molecules, separate, make them synthetic, purified, go to the pharma route. Maybe in 7 or 8 years, we will have really new treatment for sleep, new pills for sleep. But we have another route, what we call the cannabis route. And with the cannabis route, we have actually 2 products. We have one product, which is the whole extract, flowers or extract to take it to sleep. But this one, we already have a lot of data from patient and for the mice that is very useful for sleep. But we have another product. And another product, we can take just this piece, just this fraction. which still will be a cannabis oil, but without THC and without CBN, and will be very efficient to sleep. This will be cannabis oil without psychoactive action, with low side effect, but as good as the whole extract to affect you to sleep. And this is another product that I can see can come out of that. So what I showed you now, in two different studies, one on cancer, one on sleep, that today we can narrow down and define the active molecules that doing the effect that is already known in cannabis, and understand why we need few molecules and why they are working, and why we need this combination, and why actually we have one plant, one cannabis that doing this effect, and the others are not doing a specific effect, and the reason if it's have these molecules or not. So, you know, we know already that cannabis is affecting the immune cells. And because it's affecting the immune cells, we and others ask how it's affecting treating cancer with therapies that affecting the immune cells, what we call immunotherapy. Today, the major or one of the most efficient treatment to cancer, it's actually to activate the immune cells to attack the cancer cells. But if we know that cannabis is affecting the immune system, whether it's interfering or helping this treatment, this immunotherapy. And if you go to the literature, you will see that there are papers that are pro-cannabis, which showing in clinical trials that treating the patient that getting immunotherapy and using cannabis is actually more effective to them. But you can see other papers showing that it's actually interfering with the treatment. That using cannabis while you are taking, when you're using immunotherapies, will interfere with this treatment. My problem with all of these pro and con publication, that all of them coming from the same idiot, okay? All of them are publications from my lab. But the reason for that, in my opinion, it's because this patient didn't use the same cannabis that these patients are using. And for that reason, you're getting opposite results sometimes. It depends which cannabis we're using, and we can't look it on that as a global. So every one of us know that there are different apples. You know, the green one that called Smith is very sour, and the red one is sweet, and the yellow one is more flowery, okay? Every Israeli know when he's buying watermelon, to knock on the watermelon to know if it's more sweet or not sweet. We have this habit in Israel to pick the watermelon and to knock on that and to listen to that if it's sweet or not. But the meaning of that every Israeli know that every watermelon is different. Even though it's maybe from the same genetic, same plant, it will be different. But when we're going to herbal medicine, and it's not just cannabis, you can look on Archinicea, Ginkgo, we're flattening that. We said Archinicea good for the immune system. Ginkgo is good for that. Valeriana is good for reducing anxiety. But what is Archinicea? Is it one plant? Is it one species? All the Archinicea doesn't matter where it's grown, doesn't matter on which soil, doesn't matter what are the nutrients in the ground. It will be the same, have the same active compounds. And the answer is, of course, not. Cannabis is not different from other plants. Yeah, there is variety of compounds in this plant, and for that reason, we're getting different effects. What we create in my lab, and other scientists are doing that as well, we're creating a methodology to solve this problem. We can take cannabis, but also other plants, to make an extract. And I have people in my lab that expert for extract, whether this is butane, CO2 supercritical, press, ethanol, whatever. There is difference between the types of the extraction, but we are During extraction, we can analyze all the active compounds, all the cannabinoids, all the terpenoids, all the flavonoids. But if we are talking about now on a psychedelic mushroom, we can analyze all the tryptamines there. Okay, all the active compounds. Go to high throughput screening, as I showed you in the leukemia, all data from patient, as I showed you from the sleep disorder, define the effect, discover and purify the specific active compound, whether this is 1, 3, 7, or 11, we don't care. To understand the mechanism of action, go to preclinical in mice, and if all of this is working, to go to clinical study, whether with this discover purified compound or whether with the plant itself, and to get a treatment for a specific disease. We can do that in cannabis, we can do that in other plants. So what I showed you now is actually part of what we call the entourage effect, right? We're saying it's not only one molecules. Sometimes you need 3 molecules, like in the leukemia. One will bind CB1, one will bind TRPV1, one will bind something else, and we get more complex activity. So because the cannabis is harboring many compounds that are active, they can bind and do different effects. And we call it the entourage effect in cannabis. But in professional, we can call it what we call polypharmacology. And polypharmacology is when we're treating a disease by modulation more than one target. And I showed that to you in the leukemia, and I showed that you in the sleep, but I can show that in breast cancer, and I can show that in inflammation and other. And inflammation is a good example, okay? So if we're talking about this complexity or polypharmacology, it's when different compounds activate different receptors, what we call polypharmacology. If you think about it, plants, not just cannabis, harboring within this polypharmacology. If you think about treatment by a physician 10,000 years ago, maybe 3,000 years ago, and you come with inflammatory bowel disease or a pain, he won't give you a pill with one molecule. He will take a root from here, a flower from here, a leaf from here, will combine them together and will create a medicine which is actually polypharmacology. A molecule from here, a molecule from here, a molecule from here. He didn't purify this molecule, but he combined them together. For many years, the FDA, the scientist, the physician was afraid about this complexity. But what changed in the last 20 years is computers. Today, we can understand very complicated networks and how things working together using computers to understand how these 3 molecules activating together and getting this pathway. It's something that is simple today for us with the analytical tools that we have and the computer ability, what we call our bioinformatics, to combine them and to get the solutions. So this is one way to look about the entourage effect. But I want to show you that there is 3 layers, in my opinion, to the entourage effect. The first one is the polypharmacology that I just talked about. But the second one is more complicated. If we will take inflammation for an example, and I will take as an example the IBD, inflammatory bowel disease, which contain a Crohn's disease and colitis and other diseases. What's happened? Our colon cells, sometimes because of bacteria, sometimes because of other reaction, being infected. Because they're infected, they are calling our immune cells to come and to defend them, to help them. But this become a chronic reaction that instead that the immune cells will come and help, we're getting what we call kind of an autoimmune disease, that the immune cells is attacking again and again and actually harming the cells that call them to help them. So the cells secrete specific compounds called cytokines to call the immune cells to affecting them. And if we think about cannabis, in my lab, we find specific compounds that affecting the cells itself, the colon cells, to stop secreting these molecules, and by that, to reducing the effect of the T cells and to reducing the inflammation or the inflammatory bowel disease. But in different research or different study, we find other compounds that actually inhibiting the T cells or the immune cells to attack the cells. So now think about a plant, a cannabis plant, that harboring these molecules, but also these molecules. You will get a complicated action, which is stronger by using just these molecules or by just that molecules. You're getting a complicated action, which you won't get from using one molecule, so just combination. You need all of them. You need the molecules here and the molecules here. It's the same disease. We're talking about one disease, but we're affecting that from two angles to get a better action. And this is a different layer, to my opinion, of the entourage effect. So one layer was affecting one type of cells, but with few molecules that getting the effect. The second layer, it's affecting the same disease, but affecting two types of cells or two systems that getting better action. So this was the second layer. The third layer, to my opinion, is affecting different systems and affecting different things. And I will take pain as an example. My lab did till today 8 clinical trials with pain patients. And what we learned from all of these clinical trials, one of them was a big clinical trial with 3,800 patients, that cannabis is not reducing very efficiently the pain. It doesn't matter which types of pain we check, the positional pain, sensory pain, whatever pain we check, the cannabis didn't reduce this pain so efficiently. But when you questionnaire the patient, they're telling you, actually the cannabis is really, really helping them. So now we question ourselves. If the patient say that it's really helping them, you're reducing using opioids and other medicine for pain, but when you measure the pain, it's not reduced so dramatically, why it's working? And when we look at that, we saw that the cannabis, and this is not surprise, also reduce this patient anxiety, depression, help them to sleep, they are eating better, they are feeling better. And altogether, if we're checking a parameter which called catastrophism, is how the patient conceived his disease, what he's thinking about the disease, or how he suffered from the disease. From here, we see a huge improvement in the patient, what we called actually quality of life. And this, for me, is a third layer of what we call the entourage effect. Few molecules help the patient to sleep better. Few molecules reduce a little bit of pain. Few molecules make the patient reduce his anxiety. Few molecules increase his appetite. And all together, we're getting improvement of the quality of life of the patient. And sometimes one is affecting the other. If the patient is sleeping better, he will have less anxiety and less pain. If he has less pain, he probably will sleep better. But we see from different studies that there are molecules that are helping on sleep. There are molecules that are affecting anxiety. There are molecules that are affecting pain. And if you have all of them in one plane, you will get this action. And this is another layer of the entourage effect. And for that part, for the quality of life of different action of different systems that are affecting something global, I think we will never succeed to go around the real plant, the botanical plant. It's so complex, it's so many compounds on so many different systems that we're getting different action. And this is why cannabis is very unique. Cannabis is a concert of active molecules, of active cannabinoid, terpenoid, of flavonoid. But the reason that every cell in our body harboring receptors and every cell will be affected cause the cannabis not just to be a concert of molecules that are working together, but also affecting a lot of mechanisms, a lot of systems in our body that sometimes get a whole treatment or a complete treatment better than single molecule, single treatment. When a cancer patient getting chemotherapy, he suffers from pains, he's suffering from depression, he can't sleep well, he can't eat well, he have nausea, and cannabis can affect reducing nausea, increase appetite, reducing pain, have prolonged sleep, reducing anxiety. You get from one treatment, cannabis, a whole a variety of action that can really improve the quality of life. I think cannabis did a service, a huge service for medicinal plants. As I said, for thousands of years, people use plants as a medicine. Actually, most of the medicines came from molecules from plants. But in the last 100 years, when we started to create real medicines, pharma company started to patent them. Every time that they succeed to isolate one compound, just one of them, to write a patent and make a medicine, we got the medicines from a plant, like aspirin, like Taxol, like vincristine. But every time that it was more complex, there was few molecules or few action, it stayed as a medicinal plant. The cannabis opened the door, opened the window, gave scientists like myself the abilities, the money to buy the equipment, the ability to do extraction and to test it, the ability to understand the complexity of different molecules and the botanical plants on humans. And you can see that in the world starting to emerge. The biggest hospital in Israel, Ichilov, approved just 2 months ago treatment as a compassion treatment with mushrooms. The biggest hospital and the hospital gave permission to treat for compassion with a psychedelic mushroom, as we did from 2007 from cannabis. You see, it's opened the door, not just from the scientists, but also it's opened the mind of the physicians and the regulation to more complex treatment. And I think the cannabis is amazing plant, one of the phenomenal plant in the nature that harboring these molecules, but also did nevertheless revolution in the way that we will treat patients in the future. I think the future of treating diseases that we don't have a cure, that we're hitting the head to the wall years after years, like Alzheimer, like ALS, like cancer, the future will be with complex treatment as we see that with the cannabis. By that, I will finish my talk. This is the team together, my lab and my company. It's an old picture from a year and a half ago, but this is basically the team that did all the work that I just showed you. And Nirit, I think I was exactly on time, 45 minutes from beginning. So thank you very much, and I will be happy to answer any question. Nirit, if you're speaking, you're muted.
Dedi, can you hear me?
Yeah.
Someone from the organization of the ICR turned off my video. So if you, whoever did, please turn it on. Oh, okay. Here I am. Technical issues always, right? So, Dedi, first, thank you very much for this really interesting and innovative talk. I mean, the different layers of the entourage effect were fascinating. I think that we've thought about it in a different way, but never really put it terminology and looked at it from three different angles. And I think it's really helped us to see a more complete issue of the wonders of this plant. So let's go to some of the questions. We have a lot of questions. We'll try to entertain as many as we can. One question is about the variability that you showed in the beginning of the talk in the cannabis varieties. About the development of the different cannabinoid types. Do you think that we have any indications or any ability to assume or to speculate if they were developed independently? So all those chemovars. And another thing is that do we know, or do we think that the different variability aroused from a defect in the three major cannabinoids that produce the three primary cannabinoids?
So not all the cannabinoids that we analyzing are creating in the plant biosynthesis. Sometimes it's degradation or other effects like oxidation and other things in the plant. But I will say that I assume that it's at least a third of them are really enzymatically biosynthesis. So there is a diversity. I don't have any clue about how it's developed. First of all, I never thought about that, really, so I need to think about that. I can say that other plants and other species harboring cannabinoids, and sometimes it's cannabinoids that are different from cannabis, and we don't see them in cannabis. So I will assume that if it was very diverse, we will see these cannabinoids also in cannabis. The cannabis is actually limited in the types of the cannabis that we see. We don't see glycosylated cannabinoids and other modification that we see in other plants. There is a beautiful paper that was accepted just now by our colleagues in Meiri from Weizmann Institute, Asaf Aharoni, which working on a different plant and showing a variety of around 60 phytocannabinoids that none of them existed in cannabis, very potent, very active, but in the modification of the biosynthesis is different. So my feeling that the cannabis is quite narrow, like the parents, the historically parents are the quite narrow variety.
That's really interesting, and probably as you've mentioned during the talk, the fact that the evolution is not evolution, that the human induced selection was based so much on human activity and preference for the primary cannabinoids probably put a lot of emphasis on this as well.
Sure.
So this is a really good point. It's interesting. Anyways, another question is in light of the huge variability that we found in cannabis in nature, or we should probably add in terms of also part of activity of human activity, do you think, or what is the importance or how relevant are the molecular alteration methods which are used to cause variability, such as CRISPR, for example, Cas9? Do we need them?
So again, first of all, we need to understand the modification of genes changing proteins. Genes are coding for proteins, and we're talking about here about secondary metabolites, so they are not proteins. So what we can affect by this is proteins that are in the biosynthesis of these molecules. And people sometimes, people that are not biologists or physicians wrong about that. We don't know all the biosynthesis enzyme on this plant or what creating these molecules, and by that it's very difficult for us to do or to hit these enzymes, and by that today we can use CRISPR or modification just on the real major cannabinoids but we are far from understanding the what we call rare cannabinoids, or which I found in my lab, at least part of them are crucial for the effects. So we don't have this ability yet. So I'm not aware of somebody that can change or modify the plant in a way that he will create for me, the rare cannabinoids or something like that.
Right.
It's mainly THC, CBD, and CBG, and that's all.
So we are far away from this, but you're saying who knows, basically.
Yeah.
May have, yeah. So developing methods is always a good tool for hopefully it will be useful in the future. Another question takes us back to the mice experiment, with the sleep experiment that you showed us. And the question is that the focus in this experiment was on cannabinoids and isolating individual cannabinoids, or maybe combination of 2 or 3 cannabinoids that are inducing sleep. The question is, how is this related to the entourage effect, taking into consideration also other groups of secondary metabolites, such as terpenes and flavonoids?
So I show a really small part of this work. First of all, we define these 2 molecules that are really inducing sleep. When we're talking about sleep, we can use the 3 layers of the entourage effect that I said. Because first of all, sleep, when we're talking about sleep problems, we're talking about how fast you're falling asleep, how deep you're asleep, if your sleep is efficient, and how difficult for you, or you're waking up feeling alert. And these 4 parts being activated in 4 different areas in the brain, and different cannabinoids affecting different paths. So there will be molecules that will affect or will make you to fall asleep fast, but different molecules will affect your REM or non-REM going to sleep, so efficient or non-efficient. So you can get what we called this entourage effect. In the first layer, the 2 molecules affecting 1 cell and creating something, but different molecules that affecting different part of your sleep. And we can define it better regarding the problem of the patient. Second, which is very important, is this is the only indication that I found that also the terpenes are effective. So when I narrowed down and defined these 2 molecules, I didn't find their terpenes. But then after that, when I added different terpenes, I found out that there are terpenes that boosting this effect 3 or 4 times better. So, and very low amount of, so very low amount of terpenes can boost this effect. And this is what, again, connecting with what we call the entourage effect. I don't know the mechanism, I don't know why, but I can tell you for sure that there are terpenes that didn't affect at all, but there are terpenes that when you're adding that, they are boosting that effect.
Wow, so it sounds like, at least in this experiment, the terpenes were stimulating. I mean, they're not, possibly not, as you was demonstrated, they're not part of the initial response, but they're acting as synergistic.
Yeah, in a way. So you need to understand, when I'm doing the extract and treating the mice, in this extract, I have 0 terpenes because the way we're extracting and purifying and everything. But so I found out these molecules without terpenes, but then when I add the terpenes, I saw that it's very effective also. So, but alone they didn't affect the terpenes, just with the cannabinoids in this experiment. Meiri? Or you was freezing, or I was freezing, but one of us.
I'm not sure, or maybe
we're
both
back.
of us
That's
were.
what's important.
I don't.
I think Dedi, she's frozen. I think,
Okay.
yeah, Meiri.
Are we back?
She's back.
Yeah, I'm back. Is that only me that disappears, or
Yeah, it's
are
just you. It's just
Oh,
you.
wait. Okay. Another question is, can cannabinoid cause negative entourage effects? So you can take it
Yes.
as you like, as
Yeah,
interaction between.
yeah. So the answer is definitely yes. We know that CBD reduced seizures, and we tested in Israel 25 different types of cannabis, high CBD, low THC, 20 to 1. And we had 2 strains that didn't reduce seizure at all, even though they had the same amount of CBD. So when we started to look in, we defined cannabinoids that actually interfering with the effect of the CBD in seizures. So definitely, sometimes there is cases or extract that there are cannabinoids that affecting or competing with other cannabinoids on activity and can interfere with specific activity that you can look for.
No, this is very interesting.
But Nirit, it's not surprising us, right? It's a similar molecule that can compete on the same target, and by that it can reduce the effect.
That's true. It's just very difficult to isolate mechanistically and recognize mechanistically because the interesting soup, nearly 1,000 bioactive molecules that we have in this amazing plant extract. Someone else asked, would you suggest that the presence of microbiome, beneficial microorganisms on cells you are investigating can cause better effect in the entourage effect when the blend of cannabinoid is applied for a specific patient? So I think that we are dealing with...
I didn't understand the connection with the microbiome in this question. If you digest cannabis through oil or swallow cannabis, so maybe the microbiome have any kind of effect on that. If you're smoking the cannabis or vaporizing and going directly to the blood, so the microbiome is there and affecting humans, but it's not related to this section. I don't really understand the connection.
Yeah, so if we're thinking about the plant microbiome, then yes. How about, let's say, and I'm not sure which microbiome the person who asked was referring to, but let's think for a minute that we're talking about the human microbiome. So gut microbiome probably will only be involved if we're dealing with consumables.
Yeah, I guess. But gut microbiome affecting our immune and affecting our digestive and other things, but I'm not sure the connection, and the bottom line, I don't know the answer. I don't know how microbiome will affect using cannabis.
I think that no one does. So
yeah, I don't think. We tested in the trials a different side of that, how cannabis is affecting the microbiome. So how using cannabis affecting the diversity. We know that cannabis also have antibacterial or effect. And the question was, high consuming cannabis, whether it's hurting or affecting the microbiome in our body. Not how the microbiome affecting cannabis, whether cannabis is changing the microbiome. So this is something that we are testing today with the Ichilov University, or Ichilov Hospital, but I don't have the results yet.
Interesting. We'll be looking forward to this maybe next year in another webinar. Another question asked about the source of the cannabis lines that you're using. So the question is,
good answer. We will have to conclude this seminar. It looks like there's another meeting right after this. I'm so sorry.
Okay, so maybe we'll go some very, very quick last questions, and then we'll say goodbye. What is the source of the cannabis used in your research? Do you have a quality criteria for your source cannabis, such as, for example, sun-grown plants, certain known strains, grown in soil, etc.?
So, first of all, I'm looking on any source as something that can cause variability. Sometimes when the cannabis is growing bad and under stress can create different variety of cannabinoids, something that can be interesting. So this question coming from very novel place that the cannabis should be grown in a way or whatever, but as a scientist, I'm looking for diversity. In Israel, there is around 50 different authorized growers that growing many strains. I'm also working with Vulcani, with Unirit and other people in Vulcani that creating strains. This is a good source for me. I'm also working with the world. There are big companies or growers that have a variety that send it to me. I must say that in few years ago, I reached to a limit, which there is a limit of how much variety on how many strains you can hold. And today, if there is something extremely special, something that I didn't see till today, I will enter it to my bank, I will call it like that. But another high THC, low CBD or something like that, it's the variety is so big that my students can't screen thousands of thousands, they can screen how few hundreds. So I already reached there. So even if it's not extremely unique, usually I'm keeping what I have.
Yeah. Dedi, thank you so much for a really fascinating webinar and interesting, discussion. Obviously, we have this webinar raised a lot of interest and a lot of questions, and I want you to thank you very much, for your time and for sharing knowledge and thoughts.
It's my pleasure. Thank you for inviting me, and thanks for the audience for listening and for these questions. Thank you very much.
Thank you, Dedi.
Okay.
Okay, bye now.
Bye-bye.
Bye, and thanks everyone for joining us today for the webinar. And we're looking forward, well, done. Okay.
Okay, thank you, Dedi, again. I will end the webinar. See you later.